Welcome students. We'll be taking up the course on cytogenetics and plant breeding. Under the unit Alteration in Chromosome Number, the sub part of it is Aneuploidy. I am Sally Quadros-Coutinho from Government College of Arts, Science and Commerce, KPM. So, we start with heteroploidy, under which aneuploidy comes, and the types of aneuploidy. Finally, at the end of the topic, you will be able to understand the concept of heteroploidy and its types. You will be able to define what is aneuploidy and explain the types of aneuploidy with some examples. We all know every species has a characteristic number of chromosomes in the nucleus of its gametes, which are N, single set, and the somatic cells, which are normally diploid, 2N. The gametic chromosome number constitutes a basic set of chromosomes which we call as genome. And we all see here, when the male and the female haploid gametes, they have a set of chromosomes together, which we call the genome. When they come together, it results in the formation of a zygote, which helps in the formation of a diploid 2N organism. Today, we are learning about the chromosomal aberrations or mutations, under which we are going to deal with heteroploidy, wherein we see there is a change in the number of chromosomes or in the number of the set. In the previous unit, you have already learnt about euploidy, wherein the entire set changes, the number of the entire set changes in the organism. Today, we are going in depth and learning about aneuploidy. So, heteroploidy is a change in the number of a chromosome or maybe the whole set. So, that consists of aneuploidy and euploidy. Aneuploidy involves when there is the entire chromosome is less or it is extra. So, depending on that, it is further classified as hypoploidy and hyperploidy and hypoploidy. So, we know heteroploidy is euploidy and aneuploidy. And what are the effects of heteroploidy? The effects can be, there can be phenotypic changes in the organism, modification of the phenotypic ratio, alteration in the linkage groups, and some may have evolutionary significance. So, aneuploidy comes under heteroploidy. So, now we will be dealing with aneuploidy with examples. Aneuploidy is the loss or gain of a chromosome or maybe two chromosomes in a set. Now, depending upon whether it is a loss or a gain of a chromosome, it is classified as hypoploidy. Hypoploidy is when there is a loss or deletion of one or more chromosome. Heteroploidy is when there is a gain or addition of one or more chromosomes in the set. We all know aneuploidy is a Greek word which means uneven. So, when one chromosome is less in a set, it becomes uneven unit. Ploid in Greek means unit. So, in the set or unit, it is uneven, the number is uneven. So, aneuploidy. It is a karyotypic abnormality. It leads to loss or gain of a chromosome in a set leads to abnormality or variation in the chromosome number by whole chromosomes in the entire set. Normally, the genomic formula is 2N, which is of diploid organisms, 2N plus 1. This is just an example, plus 1, that is trisomy. Or if it is minus 1 from the whole set, one chromosome is missing, we call it as monosomy. N is the somatic chromosome number, which we normally see in mitotic or meiotic divisions, we talk about N and 2N, it's that chromosome number, the set. So, we learnt this. Now, we are going to learn the types of aneuploidy under hypoploidy. Hypoploidy, we see there are two types. Monosomy, where one chromosome is lacking in the, one entire chromosome is lacking in the set 2N. And depending on them, there are different syndromes that are formed or caused. Nullisomy, two chromosomes, they may be the same pair or they may be from two different pairs of chromosomes in the set, maybe missing one chromosome. So, that is called as hypoploidy, wherein less number, there is loss of chromosome in the set. So, two types, monosomy and nullisomy. Now, we go to hyperploidy. Hyperploidy means there is a gain, hyper, extra addition of chromosome in the set. If there is one chromosome added extra, then we call it as trisomy, because there are three in a pair. It should have been a pair of consisting of two chromosomes, but here there is an addition of one more chromosome. So, it is called as trisomy. And tetrasomy, 2N plus 2 chromosomes. There may be 2 chromosomes from 2 different pairs. So, like this we see there is a change in the number of chromosomes in a set, and accordingly we classify them. Now, how are these produced? How aneuploids are produced? How organisms which have less or more chromosomes produced? All this happens because of non-disjunction. Non-disjunction is when the spindle fiber may not form or it has not attached to the chromosomes, and the chromosomes remain there during the cell division. And they do not move, they go later on only in one of the gamete or the newly formed cell. The other cell may not have that 1 or 2 chromosomes or may have extra chromosome, one of the chromosomes may go the other side. So, this is called as non-disjunction. Now, let us see here in meiosis during division 1. So, here we have, suppose a diploid organism with the chromosomes. There are many chromosomes, but for convenience sake we just take the parental, the maternal and the paternal chromosomes for an example, and we see the first division. This is the first division. Now, in the first divisions we see sometimes that the chromosomes, they go only into one of the gamete. Now, in meiosis there are two divisions, the first division and here we have the second division. During the second division, we know finally there are 4 gametes formed. So, these 4 gametes formed is during the second division. During the first division, crossing over takes place in meiosis. So, at that particular time, what happens is that the chromosomes go only to one of the gamete, one of the forming cell, daughter cells. The other one does not have that chromosome, maybe 1 or 2, they are absent. The other chromosomes are present. So, what happens? Later on, when the second division takes place, this is the product what we get. So, what happens is due to non-disjunction, out of the 4 gametes, only 2 gametes will get the chromosomes, whereas the other 2 gametes will not get those particular chromosomes. The other sets are there present in all the 4, the other chromosomes of the set. So, here when there is addition, down here there should have been 1, here there should have been 1, but when there is addition of 1 more, then we call it as trisomy. If there is less 1, then we call it as monosomy. So, also we should know that aneuploids are produced or they originate in the second division also. Sometimes the first division is normal like this, see. In the previous slide, we saw that both went in one place, but here these daughter cells of the first division, the division goes normal, crossing over takes place, everything takes place, all the chromosomes go respectively in the daughter cells. Then these daughter cells again they undergo another division, second division of meiosis. At that particular time, we see that here instead of this one coming here, it goes in one gamete. So, here one gamete will not have that particular chromosome, whereas the other one is normal. So, now like this we see non-disjunction takes place and aneuploids are produced. So, we take a single one, that is monosomy. What is monosomy? Or monosomy is the formation of the aneuploid with less number of chromosome, with one less chromosome. So, these organisms we call them as monosomics. These organisms are diploid, they have all other chromosomes except one chromosome of a pair is missing. So, we call them monosomics. And these form two types of gametes. That particular organism will form two types of gametes, one with only the normal set and the other one with one chromosome less. N minus 1, that is with one chromosome less in that particular set of chromosomes. The gametes, they normally do not survive in plants. And monosomy creates imbalance due to loss of one complete chromosome. Now, sometimes we see they can be double. Double means two pairs. In two pairs there might be one chromosome missing, or even we see conditions where there is triplet monosomics, means in the set there are 3 chromosomes of 3 different pairs missing, and these missing chromosomes are non-homologous chromosomes, not of the same type or not of the same parental or this maternal and paternal, not that. So, one of that chromosome will be there, but the other one of the pair will be missing. So, like this in 3 pairs they will be missing in triple monosomics. Now, we will take an example. What happens is gametes in the plants, if they have one chromosome less, they normally die. But in man, we see the normal number of chromosomes, diploid number is 46. So, if in case of non-disjunction, if it happens, then one chromosome will be less. Total number of chromosomes instead of 46 will be 45. Now, here this is opposite to Down syndrome. We will be learning Down syndrome, but then we will further classify why we have taken this here first. So, now here this is 45. It has a serious effect where a person gets mentally retarded because of that one chromosome, means the 21st pair in the set, there are 23 pairs. So, 21st pair has one less chromosome. So, the person gets mentally retarded, remains short, shows abnormal characteristics, the tongue hangs out, and sometimes in some syndromes we see the tongue hangs out, but here in this case in Down syndrome you will see the tongue hanging out, but in this case you will see the tongue contracted, it is inside stuck up like, and the nose is prominent, ears are larger and the eyes are close to each other, whereas in Down syndrome you will see the eyes are far apart. So, what happens? The 21st chromosome, they are somatic, it is not a sex chromosome. So, this pair, the somatic pair, the 21st chromosome, instead of being in a pair like paternal and maternal, the pairing, okay, to form the diploid, here there is only one chromosome in the 21st pair, instead of being 2, it is only 1. So, that is called monosomy, and the total number comes to 45 later on. So, there is genetic imbalance which causes high mortality or reduced fertility in the organism. So, here this example we will see later on again. Now, we have nullisomy. This is again hypoploidy, which is 2n, minus 2n. 2 chromosomes are missing. So, with a pair of chromosomes missing, the organism is called nullisomic. Nullisomics often do not survive, but there is an example in wheat which is a polyploid, that is hexaploid, 6n sets, 6 sets together. That organism, if 2 chromosomes are less, it may survive, but it has reduced vigor and fertility. So, it is a nullisomic. This type, if 2 chromosomes are less, then it is a nullisomic, though it is a polyploid. So, in the sets 2 chromosomes are missing there from the 6n. Now, we go to hyperploidy, wherein addition of chromosomes. So, we take an example when there is one chromosome extra. These organisms are called as trisomics. They are diploid, but one chromosome of a single pair is extra. It can also be due to gain of 2 different chromosomes, and when it is extra, it forms a triplicate. They can be double trisomics, means in one pair 3 and another pair 3 chromosomes. So, we call it as double trisomics. So, trivalent is formed during meiosis due to non-disjunction, and this causes a lot of genetic imbalance. So, now we go to Down syndrome, which we see in man. This is an example of trisomy, wherein the 21st chromosome is affected. The person will have one extra chromosome. So, the normal is 46 plus 1, which will be total 47 chromosomes. This has a serious effect on the person, where the person gets mentally retarded, short, exhibits abnormal characteristics, tongue hangs out, whereas in the previous example monosomy, we have seen where the tongue is contracted in, the nose is flat. So, here there are 3 pairs, whereas then monosomy we saw there was only 1 chromosome. Here the pair is okay, but there is addition 1 chromosome. So, 3 together in Down syndrome. So, total is 47. Depending upon which chromosome of the complement is present in the triplicate, different types of phenotypes are produced. So, depending upon here what type of chromosome comes here as a 21st pair, 21st type of chromosome, depending upon that there are different types. So, trisomics we can call it as primary, we can call it as secondary or tertiary. So, when an organism has an extra chromosome which is identical to the homologs, just like the parental type, just exactly the same. So, same genes, same parts, everything is the same. So, when this is, we call it as primary trisomic, and the secondary trisomy, we see the extra chromosome, but a part which is genetically similar to the other half. So, isosomy. And tertiary trisomics are when the product is due to translocation. In the 3rd chromosome, there is some translocated part from another chromosome. So, that is tertiary trisomic. Then we have tetrasomy, which is 2 chromosomes are extra, and this is seen in case of wheat, wherein all 21 chromosomes can show that they can be in triplicate form or 4 chromosomes there, and such example is visible in wheat conditions are seen. Now, we have done aneuploidy, but when we have done the 21st pair, we have all seen it is autosomal abnormality, autosomes, the somatic chromosomes or the autosomal abnormalities are seen. Allosomes are sex chromosomes. So, when there is an abnormality of addition or deletion of the sex chromosomes, then there are different syndromes. We have already done Down syndrome, which is the 21st chromosome addition. So, it is the Down syndrome with 47, it is the autosomal abnormality. Whereas here the sex chromosomal abnormalities are of 2 type examples we will do today. Turner syndrome, wherein one X is less and the individual has only 45 chromosomes. Whereas the Klinefelter syndrome is having 1 extra, which leads to 47. So, see here Down syndrome is 47, but here it is autosomal chromosome, autosome, and here it is the allosome, sex chromosome is extra here. So, here also the 47 number is extra. These are aneuploids only, but they are different. Somatic, the autosomes and the allosomes, sex chromosomes and the normal chromosomes. So that makes the difference here. Now in Turner syndrome, we see there is only one X chromosome, one is less. So here we see it is like a female, sterile, short, webbing of the neck, short in height. Intelligence is not to the mark, it is less, impaired in intelligence, and they do not have their normal fertile sexual features. So the menstruation cycle is lacking there. The Klinefelter syndrome, the individuals are more male type, they look like males, but they have small, soft testes which are not developed. That is because the extra Y is here, although it would have been a female, but that one chromosome of sex chromosome being extra there, total is 47. They look like man, but they have female like enlarged breast, they are taller, obese and with less hair on the body. So we see here aneuploidy can bring about change in the phenotype. So in this topic we have learnt aneuploidy, the types of aneuploidy due to deletion and due to addition, which can be autosomal and which can be a sexual chromosomal abnormality due to aneuploidy. We have the references here, you can refer to these, and thank you.